Hello everyone. So today we are going to learn about endospore staining. In previous lecture we have seen about the endospores, how they are produced, when they are produced. So in this lecture we are going to see how we stain the endospores. So this method was started by two scientists, Schaefer and Fulton. We called it as a Schaefer-Fulton's method. We prepare a slide first, a slide of a bacterial culture. We make a smear and air dry and heat fix it. Then when our slide is prepared, then we add the first stain that we add here is malachite green stain, which is a primary stain in this case. So after adding this stain, we will keep this slide on something called the water bath, or anything which can heat our slide or heat our this smear or provide steam to our slide. We will keep this slide on that instrument. So for 5 to 7 minutes we provide the steam to this slide. So what happens in this case is we are forcing this malachite green stain to enter under the endospore, so that it will stain, or it will that stain will fix into that endospore. So after doing this for 5 to 7 minutes, this method is we use this as a mordant. In this case we are not using any extra stain as a mordant. In this case we are just providing the steam to it. So after this we will wash this slide with a water decolorizer in this case. So what happens is when we provide the steam to this smear, endospore take this stain, but the vegetative bacterial cell, because this malachite green stain is soluble in water and has not much affinity for the bacterial content or vegetative cell content, so that when we wash with water, that bacterial cell will not have this stain, but endospores will contain, or endospores will have that stain of green color. So now you can see here only endospores are stained with a green color, and we have washed because of this washing, that stain from bacterial or vegetative cell has gone already. So after that, the last step is we add another stain, which is a counter stain in this case, the saffronin, which is red color stain. So we will add this for 30 seconds, and then we will again wash this slide. So what happens is now this counter stain will stain the bacterial or vegetative cell. So now in all these methods, our endospores are stained by green color, and the vegetative bacterial cell is stained by red or pink color. Now our slide is ready to watch under the microscope. Endospores can be present in a bacterial cell in the middle, in the end. So this can be the positions of endospores. Here in this diagram you can say in some cases it is in the middle of the cell, in some cases it is in the end of the cell. So positions can differ of an endospore. So we will observe this cell under the microscope, and we can see the structure like this. This red portion is of a bacterial or vegetative cell, and this green what we wanted to observe is endospore. These green spores are, you can see that is endospore. So what is the principle behind this? So when we provide the heat, the endospores are produced by bacteria in a very harmful or extreme conditions where bacterial vegetative cell can't survive. So it is the defense mechanism of bacteria. So when we provide the heat, the normal bacterial cell will not survive, or so bacterial cell produce the endospores in this case, so that because of this action we are getting these endospores forming bacteria. Some will produce it and some will not produce it, and other bacterial cell will die. So this is the overall procedure of endospore staining. What is endospore, why they are produced, and when they are produced, that we have looked in previous lecture. So that's why I haven't elaborated on that topic. So here we are just dealing with the procedure of endospore staining. I hope that you have understood this clearly. So this is all for today's lecture. Thank you for watching my channel. You can subscribe and share, please. Thank you.